So we left off on Wednesday. I gotta remember my dates. Talking about gene expression and how it's important for development, and we're going to get into that. Okay? So gene expression patterns, we're going to get into how development is driven by gene expression in a little bit. Before we get into that, though, I want to get into another process that's also involved in development. So we talked about gene expression, important for development. Cell motility, right? Movement of cells, important for development, right? We also talked about induction, right? How cells communicate with one another. And then another process important for development, we talked about the hand, development of the limbs, all right? Digits, apoptosis, all right? So apoptosis is best described, simplest definition, programmed cell death. All right? It is an ordered pattern of how cells die. Okay? All right? And it's a triggered gene expression related way of cell death. Right? It is a very ordered pattern. All right? This occurs all the time in mature organisms, right? Cells infected, damaged, end of their functional lives, they have worn out mitochondria, whatever it might be. It's a way of the multicellular organism getting rid of worn out, dead, infected, non-usable cells without causing, say, widespread damage to other cells. All right? So here's apoptosis of a white blood cell. All right? This is a normal white blood cell to your left, right? And it has these little hang around things that kind of roll along the inside of a blood vessel, finds where it needs to be, right? But at some point, that white blood cell is no longer going to be used. Maybe it brings in a bacteria that causes death, or it maybe becomes infected by a virus, whatever it might be. All right? This is a indicative structure of a cell undergoing apoptosis. Right? What we call membrane blebbing. There's breakdown of the DNA in a certain pattern. Okay? This is just a structural example of apoptosis. Okay? You can see it much different in the membrane structure, etc. All right? So during apoptosis, DNA is broken down, and organelles and other cytoplasmic components are fragmented. All right? So basically what happens in apoptosis is there's a signal from the outside, or there's some type of stimulus that starts this sort of cascade. Enzymes get released, organelles get broken down, DNA gets cut up in what we call ladder chunks in successive chunks, about 100 base pair chunks, which eventually will be broken up. The cell becomes multi-lobed, right? Everything gets packaged up in vesicles, right? And then other cells come by and clear out the stuff that's left over. Okay? So it's this cell undergoes apoptosis, breaks things into small fragments, that gets broken up in vesicles, sort of like put out in trash bags, and then a scavenger cell comes by and cleans up all the garbage. All right? It is a protective mechanism to protect other nearby cells from further damage. All right? And this happens in cancer cells, etc. All right? But it is essential for development and maintenance in all animals, in all multicellular organisms. All right? Known to be functional in fungi and in yeasts, right? But in vertebrates, what is it important for? Nervous system, right? But probably the best known example of development for apoptosis is the formation of the limb, of the digits, hands and feet. Okay? Because what occurs during normal is you have limb bud formation, right? And limb buds are projections off the embryo, right? They end up growing out like sort of like this. This is a emerging limb bud. And this will end up becoming, digit 1, 2, 3, 4, and 5. Those sort of limb bud patterns off the top there. All right? But the interdigital tissue, the cells in between where those digits will be, will undergo apoptosis and they'll regress. All right? And forming space between digits. And those of you who know people with webbed feet, all right, at some point, they may not have had a portion of this here grow out or undergo complete apoptosis, and you end up with that. So that part of their fingers are sort of stuck together. Now that's easily surgically repaired as a child, right? Where you just snip the tissue and then it's right. That's one way of it occurring. All right? But apoptosis is important for that function. Okay? For webbed feet where it's just the skin, that's an easy surgical procedure. But changes in the molecular biology of this and the gene expression of this can actually cause fusion of the digits and then disruption of the limb buds or the digital formation in general. So this is meant to illustrate the importance of apoptosis in development. So what processes do we overall have to talk about in terms of development? Gene expression, cell communication, example being induction. All right? We have cell motility, all right, movement of cells from one area to another, and apoptosis are all important cell processes that drive development. All right? So how do we study development now? All right, so those are the processes that are involved in developing of multicellular organisms. We talked about how we would study development previously. What do we need? We need a model organism, right? Yes? All right. So one of the model organisms is a fruit fly. We're going to go through that in a little bit. All right? So we're going to use the fruit fly, Drosophila, as an example. So you'll understand sort of the background of that. All right? But one of the first things that we did when I showed you the frog egg, what did it look like?
Half and half. It was like half white, half gray.
All right, so here's the egg. All right, it looks sort of like this, right? And what was important about having the dark area up top and the white area at the bottom? Separate the egg and the yolk. And what happened when I showed that video? What happened to the gray part? What was its fate?
Tadpole.
It grew into the actual animal, right? That part, and the white part ended up becoming the yolk and wasn't part of the animal, right? So from the very beginning, how the egg is formed, you have a difference in top and bottom, right? And eventually you have a difference from front to back, right? So this is what's called pattern formation. All right? At the very beginning of development, because the egg ends up being a sphere, right? No front, no back, at least from its general shape, right? No front, no back, no top, no bottom. And it's floating around in space, can't determine what's what. So there's elements within the egg itself that will help determine front, back, top, bottom, left, right. This is pattern formation, and then this leads to what we call axis determination. All right? So pattern formation, development of a spatial organization of tissues and organs. It's dependent upon knowing what's top, what's bottom, what's left, what's right. All right? So you have that positional information which is determined by molecular cues. So what within the egg will help determine what's front, what's back, what's left, what's right, what's top, what's bottom? Gene expression, those cytoplasmic determinants that have been produced, right? Those transcription factors that are present on one side and a higher population on one side than the other. And during cleavage, right, those cell divisions that occur, you end up with different cells that now become front to back. All right? So those molecular cues in these could be, right, example, those cytoplasmic determinants. Okay? This ends up giving you your relative position for body axes. This is axis formation. All right? And so the pattern formation was the first and best described using the fruit fly as an example. Okay? What's the advantage of using the fruit fly as a genetic organism to study in the lab? What's that? They reproduce fast. All right? Typical generation time is about a day. Right? They live for about, so you can have multiple generations of a strain of fruit flies in a week. What are other genetic organisms that are used besides the fruit fly? Mice. All right, let's compare mice to fruit flies. Mice become sexually mature in about 3 to 4 weeks. So from birth to about 3 weeks before they can have the next set of litters. All right? So already you're talking about one generation in 3 weeks, 4 weeks, right? Before you can have the next generation. Whereas in Drosophila, you've probably had 12 generations in that time. Okay? All right? What else about mice is problematic? You have to keep mice in a certain housing. Keep it pathogen-free. Right? They have to be genetically monitored. Right? Drosophila, you can set it up in the lab. There's no determination of how you care for those mice or how you care for those fruit flies. You can do whatever you want to them. The bureaucracy dealing with them is much easier. Okay? So Drosophila is an ideal genetic model organism. All right? So combining sort of the anatomical, genetic, biochemical approaches, using Drosophila as a model organism, they've discovered developmental principles common to many other species. Well, that's the other thing. Drosophila, although an invertebrate, an insect, does have many genes that are conserved and applicable to humans. All right, and you'll see that in a little bit. All right, so the life understanding sort of how they did this, you have to understand the life cycle of fruit flies. All right, so fruit flies have a modular structure. Right, so the fruit flies are arthropods; they're insects. They're composed of they're segmented. Right, they have segmented bodies, and you'll see this in terms of the larval stage that you look at. They are patterned. Right, so that you have segment 1 through 5, right, 6 through 13. Every embryo of a Drosophila has about 13 segments. Okay, part of which becomes the head, the middle part becomes the thorax that has the legs, and then you have the abdomen, which is the posterior portion, right, that has where those legs and wings are is defined on the thorax versus antennae that are on the head, eyes, etc. All right? So you have an idea of where things are supposed to be. All right? And so in Drosophila, just similar to what we talked about in the frog egg, we use the frog egg as an example, cytoplasmic determinants, mRNA, transcription factors, etc., right, in the unfertilized egg determine the axis before fertilization. So a protein, an mRNA that is present in the egg prior to fertilization already tells which is the head or the anterior portion and where the posterior portion is. Okay? So here's the adult fruit fly head, okay? And you'll see that the colors, green, brown, and orange, are meant to show you where the head comes from in terms of the larval segments, okay? So on the right, you have the development of the egg. So you have nurse cell, a nucleus, the egg itself develops, right? Here's your depleted nurse cell, your eggshell. This is the fertilized egg, all right? So it already has sort of a setup from there, okay? And then you end up having embryonic development. Here are your body segments. Segmented embryo, you see you have roughly 13 embryos and you get into the larval stage, all right, before morphogenesis occurs. All right, those first two segments become the head. You have the next 4 or 6 that become the thorax, and then the final sections become the abdomen. All right, and in the mature organism, the head has a certain structure with antennae, the thorax has a certain structure with wings and legs, and the abdomen has a certain structure. All right? All right, so the Drosophila eggs develop in the female's ovary. After fertilization, the development results in segmented larva. Eventually, the larva forms a cocoon within which the metamorphosis turns into an adult fly. Okay? So pretty straightforward. You understand the development of, in general, understand the development of a fruit fly from fertilized egg to mature organism. And that the segments are important for where they're going to become, what they're going to become eventually. Right? So how do these things, or how do they know this? All right, so here's your segmented. All right. So, Lewis, Vollard, and Weishaus won a Nobel Prize in 1995 for decoding pattern formation in Drosophila. All right? Lewis discovered something called a homeotic gene. All right? But they control pattern formation in the embryo, the larva, and eventually the adult. Okay? So, remember we have head, thorax, and abdomen portions. Okay? And how does the embryo know that the first two segments become the boundary between segment 2 and 3? Is that important?
Yes.
Yes, because that defines the beginning of the thorax and the end of the head, right? So knowing what segments are where is important in the overall development of the mature organism. And changes in that can lead to some funky things. All right? So what you see here is the wild type on the left, the normal fruit fly head. Eyes, antenna, okay? This is a scanning electron micrograph of the normal fruit fly head on the left. What's different on the right?
They have legs on their face.
They have legs on their face. They have legs where antennae are supposed to be. That's not normal. Okay? What has happened? What has happened? A mutation. A mutation in what? In gene expression, in its face, yes. Okay? A mutation in gene expression that has changed what? Has changed the pattern. So now that segment 3, what has occurred? The boundary between where the head's supposed to be and where the thorax is supposed to be is changed. So that now part of where the legs are supposed to be are on the head. Okay? You've changed sort of where things are meant to go. All right? So this is where they study segment formation. Sarah?
Can the flies live like that, or do they die?
They live like that. They have disrupted function because the antennae have function in terms of inputs in the nervous system and feeling around and
Do they like run into things?
yeah, they're not. And there are thousands of mutations that are involved in this. Okay? So what they did was they created mutants, conducted breeding experiments, and they looked for corresponding genes. All right? So they looked at these mutations in certain genes, and a lot of them are embryonic lethal, which means what? They don't produce mature organisms, they die in the embryo. All right? Causing death during embryogenesis. But they found 120 genes that were important in segment formation. All right? So for normal segmentation, so they have 120 genes that are important for defining what's the head, what's the thorax, what's the abdomen, what's the first segment of the thorax as opposed to the last segment of the thorax. Okay? What is important for the determination between the end of the head, the beginning of the thorax, the end of the thorax, beginning of the abdomen. All right? And whether or not you could shorten the abdomen, right? The length of it. All right? So 120 genes that determine, say, they developed mutants that had wings on their head, right? No wings, two sets of legs as opposed to 3, extended abdomens, etc. All right? But it's all important for segmentation. All right? And this is important for axis determination. Okay? So axis determination, what determines a pattern and an axis in the Drosophila embryo? Cytoplasmic determinants. Okay? And where do those cytoplasmic determinants come from? They're present where? They're in the cytoplasm of what, Drew?
Cell.
What cell? The egg. And so the egg then is produced by the mother. So these are what we call maternal effect genes. Okay? So the genes, they're looking at all these different mutants, and some of the mutants are important for pattern formation, okay, axis determination, and they are maternal effect genes, meaning they come from mom. They originate from mom during the formation of the egg. All right? And then you can further break these maternal effect genes down. All right? They're also called egg polarity genes. All right? So really, we're talking about cytoplasmic determinants come from the mom, and then they determine egg polarity, front to back, left to right. They control orientation of the egg, and then consequently, the fly. Questions on that? One of these genes, we're going to go through one of these genes. All right, it's called bicoid. Okay, they named it, I don't know. All right, so bicoid is a maternal effect gene. So bicoid's the protein. All right, comes from the bicoid gene and affects or determines the front half of the body. Where this gene is expressed, where this protein is found, determines the head. What's the head, what's the back? Okay? So, an embryo whose mother, so they developed a mutant where the mom no longer produces bicoid. All right? Now the embryo from the mother who doesn't have any functional, lacks the front half of its body and has duplicate posterior structures at both ends. So what does it look like? Basically, it's two abdomens. All right? It doesn't develop a head. It doesn't develop a head. So it ends up being something in terms of the larva, right? It ends up having, segments like that. It doesn't have a defined head segment versus a abdominal segment. All right? So this is what it looks like. Okay? What you see in the top is normal segmented embryo. Head, 3 thorax segments, and then 8 abdominal segments. Okay? Now, with the tail. Now with the mutant larva for bicoid, there's no bicoid present. What happens? What conclusion can you make from this?
It's gonna die. It's not gonna live.
Well, it's not going to live. Okay. Why isn't it going to live?
Doesn't even have.
There's no development of a head, right? There's no development of a thorax. What about the abdominal segments? Look at the numbers of the abdominal segments up there.
It's weird. I can't read it.
This is
Oh wait, I have.
8, or say 8, 7, 6, 7, 8. And then there's a tail here and a tail there.
There's only 1/6.
All right. Which is the absolute posterior portion,
6
right? So the absolute posterior portion of the normal embryo. Okay? So basically, like Ben said, it has 2 butts. Right? There's nothing else that's developing because no bicoid is present. So what do you make a conclusion from that? What conclusion can you make from that? Bicoid is necessary for posterior development. Okay? Yes? Or for anterior development. You all right? For the development of the head. Bicoid is absence is then necessary for posterior development. What would you think is going to happen if I had an embryo and I added bicoid mRNA from the outside to the posterior to the egg on the opposite end? It's going to have 2 heads, right? And a thorax in the middle. Why? Because now I have too much bicoid at both ends. The absence of bicoid is important for the development of the posterior portion. Okay? So if you have bicoid on both ends, it's going to end up causing a head-head formation. All right? So this suggests that the product of the mother's bicoid gene is concentrated at the future anterior end and is required for setting up the anterior end of the flock. All right? Bicoid is what we call a morphogen. Okay? Why? What does it stimulate? Morphogenesis. It stimulates body patterning, right? It is also dependent upon a gradient. Okay? Where is the highest amount of bicoid going to be found? At the head. Where is the lowest amount of bicoid going to be found? At the tail. So if the absolute highest amount of bicoid is found at the head, right, that determines segments 1 and 2. A little less is found in segments 3, 4, and 5. Right? 3 greater than 4, greater than 5. And that sort of middle range determines the thorax. A little lower is the beginning of the abdomen. Right? So it depends upon concentration also. Go from high to low, and you now determine segments 1 through 8. Okay? So, the bicoid mRNA is highly concentrated at the anterior end of the embryo. After the egg is fertilized, what's going to happen to that mRNA? mRNA is present, eggs fertilized, now translation is going to start. So now you're going to produce bicoid protein. All right? That So that then starts to produce genes or induce transcription of genes that are important for head formation. All right? But like I said, if you inject bicoid mRNA into various regions of an embryo, it results in the formation of anterior structures at the site of injection. Right? So you can develop the head structure on, say, the abdomen. All right? That's an animation. Okay, so here's sort of the results. Here's your bicoid mRNA in the mature unfertilized egg. All right, and this is, so you have the actual results up top, right, picture from up top, and then you have the sort of graphical representation of that. Fertilization, translation of the bicoid mRNA. Here's your bicoid protein in the early embryo, showing your, so the bicoid mRNA is on one side, and it sort of forms a concentration gradient across the side. Why is this how is this going to work? If I'm talking about, there's a bunch of cells in here, how does this work across different cells? So what's unique about this is that this embryo forms what's called a syncytium. All right? It basically means that there's a bunch of cells inside this, but that they share cytoplasm among them, so that transcription factors can be shared among different cells, so that this gradient can be formed. Okay? So it's not just one cell. There's actually multiple cells, but it ends up forming a pattern or a gradient. All right, so then you have your bicoid protein in the early embryo, which then determines your head. All right, why is this important? All right, it identified a specific protein required for some early steps in pattern formation. All right, so bicoid ends up becoming a transcription factor, high point, like where it's high, where it's present, it determines or the expression of certain homeotic genes, right, which then tells it's a head formation versus thorax, etc. All right, it helped increase the understanding of the mother's role in development. Right, the fact that mom developing the egg is going to influence what happens in the embryo during development, even before fertilization. There's already some type of pattern established. All right? And this then finally, there was the gradient principle, molecules that can determine a gradient of molecules that can determine polarity and position in the embryo. All right? Since this bicoid was produced, then they looked at other possibilities of morphogens, and they found that bicoid is one, and there's another one that determines posterior, right? And so you have posterior morphogen determines the end, and an anterior portion, and they have opposite gradients. So that in the middle they're about equal, right? And then one's high on one side and low on, and low on the other, and then vice versa, so they're sort of inversely proportional, all right? But the proportions are important for determining anterior-posterior pattern formation. Questions on bicoid? Formation or anything about Drosophila? Now, after we come back from break, we'll continue with this, all right, and how homeotic genes are important for determining, segment head, thorax, abdomen, and changes in them. And I will also tell you that mutations in homeotic genes are important for a lot of other genetic mutations in humans as well. All right, we have analogs of homeotic genes called Hox genes that are important for determining what's what.